Good day, this is Professor Will, CBMD, PhD. Today is Sunday, May 27, 2018, and it's 6:07 and 57 seconds a.m. Eastern Standard Time. Gut bacteria play key role in anti-seizure effects of ketogenic diet. Date, May 24, 2018. Source, University of California, Los Angeles. Summary, scientists have identified specific gut bacteria that play an essential role in the anti-seizure effects of ketogenic diet. The study is the first to establish a causal link between seizure susceptibility and the gut microbiota, the 100 trillion or so bacteria and other microbes that reside in our intestines. The ketogenic diet has numerous health benefits, including fewer seizures for children with epilepsy who do not respond to anti-epileptic medications. However, there has been no clear explanation for exactly how the diet aids children with epilepsy. Researchers in the laboratory hypothesized that the gut microbiota is altered through the ketogenic diet and is important for the diet's anti-seizure effects. Research team conducted a comprehensive investigation into whether the microbiota influences the ability of the diet to protect against seizures, and if so, how the microbiota achieves these effects. In the study of mice as a model to more thoroughly understand epilepsy, the researchers found that the diet substantially altered the gut microbiota in fewer than 4 days, and mice on the diet had significantly fewer seizures. To test whether the microbiota is important for protection against seizures, the researchers analyzed the effects of the ketogenic diet on two types of mice: those reared as a germ-free in a sterile laboratory environment, and mice treated with antibiotics to deplete gut microbes. In both cases, we found the ketogenic diet has no longer effective in protecting against seizures. This suggests that the gut microbiota is required for the diet to effectively reduce seizures. The biologists identified the precise order of organic molecules known as nucleotides from the DNA of the gut microbiota to determine which bacteria were present and at what levels after the diet was administered. They identified two types of bacteria that were elevated by the diet and played a key role in providing this protection. With this new knowledge, they studied germ-free mice that were given these bacteria. We found we could restore seizure protection if we gave these particular types of bacteria together. If we gave either species alone, the bacteria did not protect against seizures. This suggests that these different bacteria perform a unique function when they are together. The researchers measured levels of hundreds of biochemicals in the gut, blood, and hippocampus, a region of the brain that plays an important role in spreading seizures in the brain. They found that the bacteria that were elevated by the ketogenic diet alter levels of biochemicals in the gut and the blood in ways that affect neurotransmitters in the hippocampus. How do the bacteria do this? The bacteria increase brain levels of GABA, a neurotransmitter that silences neurons, relative to brain levels to glutamate, a neurotransmitter that activates neurons to fire. This study inspires us to study whether similar roles for gut microbes are seen in people that are on the ketogenic diet. The implications for health and disease are promising, but much more research needs to be done to test whether discoveries in mice also apply to humans. Good day, this is Professor Will, CBMD, PhD. Today's Sunday, May 27, 2018. It is 6:21 and 59 seconds a.m. Eastern Standard Time. New method for finding disease susceptibility genes. Date, May 26, 2018. Source, National Institute of Science and Technology. Summary, a team of researchers has unveiled a novel statistical algorithm capable of identifying potential disease genes in a more accurate and cost-effective way. In the study, the research team presented a novel method and software GSA SNP2 for pathway-enriched analysis of GWAS p-value data. According to the research team, GSA SNP2 provides a higher power decent type 1 error control and fast computation by incorporating the random set model and SNP count adjusted gene score. GSA SMP2 is a powerful and efficient tool for pathway enrichment and network analysis of genome-wide association study GWAS summary data. With this algorithm, we can easily identify new drug targets, thereby deepening our understanding of disease and unlock new therapies to treat it. Each individual's genome is a unique combination of DNA, a sequence that plays a major role in determining who we are. This accounts for all individual differences, including susceptibility for disease and diverse phenotypes. Such genetic variation among humans are known as single nucleotide polymorphisms, SNPs, that correlate with genetic specific disease, could serve as predictive biomarkers to aid the development of new drugs through the statistical analysis of GWAS summary data. It is possible to identify the disease associated with SNPs. Despite the astronomical amounts of money and time invested in the statistical analysis of SMP data, the conventional SMP detection technologies have been unable to identify all possible SMPs. This is because most of the conventional methods for detecting SMPs are designed to strictly control false positives in the results. Therefore, among tens of thousands of genomics data and hundreds of thousands of SMP analyzed, the number of markers described within a candidate disease gene often reaches several tens. Although controlling false positive SMPs is needed for the correct interpretation of the result, too much filtering may hamper its usefulness in drug development. Therefore, enhanced statistical power is essential to practical statistical algorithms. The team aimed to develop an algorithm that improves the statistical predictability while maintaining accurate control of false positives. To do this, they applied the monotone cubic spline trend curve to the gene score via the competitive pathway analysis for gene expression data. In a comparative study using simulated and real GWAS data, GSA-SMP2 exhibited higher power and best prioritized gold standard positive pathways compared with 6 existing enrichment-based methods and 2 self-contained methods. Based on these results, the difference between pathway analysis approaches was investigated and the effects of the gene correlation structures. The pathway enrichment analysis were also discussed. In addition, GSA-SMP2 is able to visualize protein interaction between networks within the cross-significant pathways so that the user can prioritize core subnetworks for further study. According to the research team, GSA-SMP2 provides a greatly improved type 1 error control by using SMP count-adjusted gene scores. While nevertheless preserving high statistical power, it also provides both local and global protein interaction networks in the associated pathway and may facilitate integrated pathway and network analysis of GWAS data. The research team expects that the GSA-SMP2 is able to visualize protein interaction networks within across the significant pathways so that the user can prioritize the core subnetworks for further study. This is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018. It is 6:42 and 39 seconds a.m. Eastern Standard Time. Science News. New chromosome study can lead to personalized counseling of pregnant women. Date, May 25, 2018. Source, University of Copenhagen, the Faculty of Health and Medical Sciences. Summary, fetuses with specific rare chromosomal aberration have a 20% risk of a developmental disorder or another brain disorder. A new study reveals. The findings may provide personalized diagnostics and counseling for these pregnant women. These chromosomal aberrations are seen in the fetuses in 1 out of 2,000 pregnant women. Until now, when such an aberration has been found, the medical doctors have told the pregnant women that the fetus risk of developing congenital malformations is 6 to 9%. We have identified all the people who as foetuses were diagnosed with such a chromosomal aberration in Denmark, and we can see that they more often have developed the disease. The previous study, which have found a risk of 6 to 9%, mainly looked at congenital malformations and did not include neurocognitive disease such as autism and mental retardation, which often emerge at a later stage. Chromosomal material that has been exchanged. We call these aberrations balanced chromosomal rearrangements because all the genetic material is still there. There is neither a loss nor a gain of genetic material. The problem is that parts of genetic material have been exchanged and that might have caused disruption of an important gene. The study is the largest systematic survey of these rare chromosomal aberrations in foetuses, and it also evaluates the methods that can be used for examining them. These chromosomal abnormalities are diagnosed through chorionic villus sampling and amniocentesis by classical chromosomal analysis, where the genetic material is examined in a microscope. This method has been used for the last 40 years, and it is still the method used in most pregnancies globally. However, in Danish hospitals, the method is increasingly being replaced with another method, chromosomal microarray, which exclusively tests for less or gain of the genetic material. Chromosomal microarray therefore cannot discover the rare balanced aberration studied here. In contrast, the study reveals that modern genome sequencing in most cases will be able not only to detect these balanced chromosomal aberrations, but also show whether genes have been damaged. The study is a collaboration between researchers at the University of Copenhagen doing basic research at the Clinical Genetic Department of Denmark and Department of Epidemiological Research Statens Serum Institute. The researchers carefully examined the Danish health records to find everyone born with a de novo balanced chromosomal aberration since 1975. Each time they found a person with the aberration, they established a control group, of 5 individuals with normal chromosomes who had been born more or less at the same time by a mother of the same age. The researchers then visited the majority of the persons to do a health examination and collect blood samples for modern genetic analysis by comparing the health data of the group with the chromosomal aberrations in the control group. The researchers found 2-3 times higher risk of developing a neurocognitive disorder in the group with the chromosomal aberrations. In addition, the researchers found that new whole genome sequencing techniques are much better than any other techniques at assessing the health effect of a balanced chromosomal aberration. This is the conclusion after almost identical assessments made by 2 independent research groups, including a group from the Harvard Medical School. We have not known which analysis to use to discriminate the foetuses that will be healthy from those that will eventually develop disorders. Our study shows that by using the new sequencing technologies, we can in fact discriminate in a number of cases. This may greatly affect the diagnosing and counseling of pregnant women carrying foetuses with this specific type of chromosomal aberration in the future. Good day, this is Professor Will, CBMD, PhD. Today is Sunday, May 27, 2018. It is 7:26 and 58 seconds a.m. Eastern Standard Time. Science News. Potent new mechanism of actions for treatment of inflammatory bowel disease. Date May 25, 2018. Source University of TS UK UBA. Summary. Through research on the small molecule analog of E6007, which is under clinical development as a treatment for inflammatory bowel disease, a novel mechanism of action was revealed in which this analog inhibited the adhesion and infiltration of various leukocytes through the blockade of certain interactions. IBD refers to a group of intractable disease which lead to repeated inflammation in the mucus of the large or small intestines, resulting from an unidentifiable cause. According to a survey by Japan Interactable Disease Information Center in 2013, IBD had the greatest incidence among young people in their 20s and 30s. Among IBD, it is reported that the number of patients with UC ulcerative colitis and CD Crohn's disease was 166,060 and 39,799 respectively, which means IBD is the intractable disease with the greatest number of patients. Currently, in addition to observing infiltration of various leukocytes in the inflammation sites of IBD, ITGA4 is strongly expressed, and therefore treatment consists of leukocyte apheresis, therefore or monoclonal antibody treatment targeting ITGA4. However, with the number of IBD patients increasing year after year, development of small molecule treatment that makes it easy to comply with treatment and has superior efficiency is highly anticipated. As a new IBD treatment with a mechanism of action for inhibiting integrin activity and using an analog of this E6007ER464195-01, the Joint Research Group utilized a biomarker technology developed at the University of TS UK UBA, which visualizes protein-protein interaction in an attempt to reveal the mechanism expressing anti-inflammatory effects. And CRT, a molecule chaperone, binds to integrin subunits and promotes cell adhesion, using a biomarker to investigate CRT and ITGA4 interaction in the colonic structure of UC patients. The Joint Research Group found that interaction of inflamed sites significantly increased compared to healthy areas, given that dissociation of CRT and ITGA4 interaction could suppress activation of leukocytes. High-throughput screening assay was conducted on consequently ER46419501 was identified as a small molecule that suppresses leukocyte adhesion by binding to CRT and inhibiting CRT-ITGA4 interaction. When mice were orally administered ER464195-01 as a prophylactic treatment, in addition to exhibiting remarkable anti-inflammatory effects, indextrin sodium sulfite DSS induced colitis from comprehensive analysis of gene expression using RNA sequencing found that inflammatory cytokines and expression of inflammatory response signaling factors were significantly suppressed. Furthermore, when ER464195-01 was therapeutically administered to mice with DSS-induced colitis, it was interesting that mucosal barrier injury as well as infiltration of inflamed cells were remarkably improved. This novel mechanism of action revealed through the joint research is expected to lead to the provision of a new IBD treatment option. ER464195-01, which possesses the mechanism of action revealed through this joint research, and an analog E6007, and it is believed that E6007 also has the same novel mechanism of action. Therefore, our finding is expected to lead to value enhancement and acceleration of the development of E6007, which aims to provide a new treatment method for IBD patients. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018. It is 7:37 and 02 seconds a.m. Eastern Standard Time. Some veterans at higher risk of Zika complications. Date, May 24, 2018. Source Plus. Summary: Zika virus, ZIKV, has affected roughly 500,000 people in Western Hemisphere in recent years, including U.S. veterans, older veterans, and those with comorbidities are at increased risk of hospitalization and neurological complications after ZIKV infection, researchers now report. ZIKV is a flavivirus transmitted primarily by Aedes mosquitoes, and outbreaks spanning 2015 and 2016 affected hundreds of thousands of people, including more than 5,000 cases in the continental U.S. and more than 36,000 in Puerto Rico and in the Virgin Islands. The Veterans Health Administration, VHA, has healthcare facilities throughout the U.S. and territories and performs ongoing surveillance for emerging pathogens. Scientists performed a case series study of all ZIKV cases in VHA hospitals in 2015 and 2016. They analyzed the patient characteristics and clinical outcomes of 736 ZIKV positive patients out of the 1,538 patients who were tested for ZIKV during this time period. 736 48% of the patients tested positive for ZIKV, 655 89% were male, and 683 93% were seen in the VA Caribbean healthcare system. 94 and 13% of the patients were hospitalized, and 19 and 3% died after their ZIKV infection. In general, veterans who were older and had other underlying health problems, including congestive heart failure, dementia, and acute kidney injury, were more likely to be hospitalized. If a patient presented with a rash, they were less likely to be hospitalized. But if they presented with initial neurological symptoms, they were more likely to be hospitalized. Better understanding of those patients most at risk for severe disease can help providers when evaluating and treating patients with ZIKV infection. Good day, this is Professor Will, CBMD, PhD. Today is Sunday, May 27, 2018. It is 7:47 and 11 seconds a.m. Eastern Standard Time. Chemists develop new blood tests to quickly detect liver damage. Date, May 24, 2018. Source: University of Massachusetts of Amherst. Summary: chemists have developed a quick and robust blood test that can detect liver damage before symptoms appear, offering what they hope is a significant advance in early detection of liver disease. Their new method can detect liver fibrosis, the first stage of liver scarring that can lead to fatal disease if left unchecked, from a blood sample in 30 to 45 minutes. Their new method can detect liver fibrosis, the first stage of liver scarring that can lead to fatal disease if left unchecked, from a blood sample in 30 to 45 minutes, the authors note. They point out that liver disease is the leading cause of premature mortality in the United States and UK, and is rising. It often goes unnoticed until late stages of the disease when the damage is irreversible. IALS designed a sensor that uses polymers coated with fluorescent dyes that bind to blood proteins based on their chemical processes. The dyes change in brightness and color, offering a different signature of blood protein pattern. This platform provides a simple and inexpensive way of diagnosing disease with potential for both personal health monitoring and application in developing parts of the world. Clinics and hospitals to screen people with elevated liver disease risks so they can be treated before it is too late. The UCLA team tested the sensor by comparing results from small blood samples equivalent to fingerprint checks from 65 people in 3 balanced groups of healthy patients, and among those with early stage and late stage fibrosis, which was determined using the Enhanced Liver Fibrosis ELF test, the existing benchmark for liver fibrosis detection. They found that the sensor identified different protein level patterns in the blood of people in the 3 groups. The ELF test requires samples to be sent away into the lab. By comparing the different samples, the sensor array identified a fingerprint of liver damage. It is the first time this approach has been validated in something as complex as blood, to detect something as important as liver disease. The investigators report that the test distinguished fibrotic samples from healthy blood 80% of the time, reaching the standard threshold of clinical relevance on a widely used metric and comparable to existing method of diagnosing and monitoring fibrosis. The test distinguished between mild, moderate fibrosis and severe fibrosis 60% of the time. The researchers plan further tests with larger samples to refine the method's effectiveness. These exciting findings epitomize the mission of IALS to translate excellent basic science with diagnostics, therapeutic candidates, and personalized health monitoring devices to improve human health and well-being. This may open the door to cost-effective regular screening program thanks to its simplicity, low cost, and robustness. We are addressing a vital need for point-of-care diagnostics and monitoring, which could help millions of people access the care they need to prevent fatal liver disease. Scientists state the sensing strategy uses a signature-based approach that is highly versatile and should be useful in other ways. A key feature of this sensing strategy is that it is not disease-specific, so it is applicable to a wide spectrum of conditions, which opens up a possibility of diagnostic systems that can track health status, providing both disease detection and monitoring wellness. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, is 9:10:02 a.m. Eastern Standard Time. Science News: Molecular network boosts drug resistance and virulence in hospital-acquired bacterium. Found in Acinetobacter baumannii, this network could potentially be targeted to fight infection. Date May 24, 2018. Source Plus. Summary: in response to antibiotics, the gene regulation network found in the bacterium Acinetobacter baumannii acts to boost both virulence and antibiotic resistance. Baumannii is a multi-drug resistant bacterium that spreads in hospitals around the world, causing serious infections such as pneumonia, wound infection, and sepsis. Previous research has shown that the bacterium's BF-MRS system, a network of molecules that influence the gene expression, enables it to build a protective capsule around itself in response to attack with antibiotics. Despite the importance of BF-MRS, its precise role in regulation of gene expression in the bacterium and the resulting influence on the ability of A. baumannii to resist antibiotics and cause disease have been unclear. Scientists performed a series of experiments to clarify the role of BF-MRS. Using strains of A. baumannii that were genetically altered to either activate or wipe out the BF-MRS stress response, the researchers showed that BF-MRS enables lethal sepsis in mice. They also tested how these strains responded to antibiotic exposure, finding that the BF-MRS stress response confers resistance to a wide variety of drugs. The research team also showed that these effects are connected to the genetic influence of BF-MRS, demonstrating through RNA sequencing experiments that it can reprogram gene expression throughout the A. baumannii genome. In particular, BF-MRS controls a variety of gene pathways involved in construction and integrity of multiple components of the cell envelope, as well as cell division. In the final experiment, the scientists showed that mutations to enhance antibiotic resistance could bypass the need for BF-MRS and an A. baumannii strain in which BF-MRS had been wiped out. These mutations also impacted processes that influenced the cell envelope and division, highlighting the link between drug resistance and cell physiology. Together, these findings show that BF-MRS plays a central coordinate role in the intertwined ability of A. baumannii to resist antibiotics and boost virulence in response to them. This suggests that inhibition of BF-MRS could serve as a potential strategy to combat A. baumannii. And we revealed that a single two-protein system controls a global network of proteins that is critical for making A. baumannii a threat. These proteins control broad drug resistance and the potential to cause opportunistic disease, and central on processes that build and maintain the cell envelope. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018. It is 9:31:15 a.m. Eastern Standard Time. Science News: Programming synthetic molecular codes to turn genes on. Scientists developed a biometric epigenetic code that can activate genes on demand to treat disease. Date May 24, 2018. Source: Coyote University. Summary: a team of researchers developed a synthetic molecular code to script gene activation. The process could help lead to future gene-based therapies for a wide array of diseases. In particular, the code could help combat epigenetic mutations which change how genes express themselves and can play a critical role in neurodegenerative disorders like Parkinson's disease, Alzheimer's disease, and multiple sclerosis. Scientists fabricated a molecular code that mimics a key process that turns on genes in the body. The code targets histones, the proteins that are responsible for packaging DNA so that it fits inside the cell's nucleus. If you uncoil the DNA in a single cell, it would be about 2 meters long. To fit inside cells, DNA is tightly wrapped around histones. When histones undergo a chemical process called acetylation, an acetyl group is added to a part of their structure. This loosens DNA attachment to their proteins, which leads to gene activation. Scientists have been researching ways to influence histone acetylation in order to manipulate gene activation, but current methods have their shortfalls. For example, some synthetic molecules are easily degraded by enzymes in the body. Others are inconsistent in their ability to activate genes. Scientists developed a molecular program that recruits a histone acetylating enzyme to a specific part of the DNA strand. The program, called PIP, is formed of 2 components: a bromodomain inhibitor, which recruits a specific type of histone acetyltransferase enzyme, and a synthetic hairpin-shaped molecule that recognizes a specific DNA sequence. The code was successful in emulating the natural histone acetylation process and led to the activation of specific gene associated with central nervous system inside the living cells. However, the researchers note that further work is needed to improve PIP gene selectivity. This work adds to a library of small molecular genetic regulators that could form the basis for epigenetics and future gene therapies to treat multifactorial neurodegenerative disorders. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, 9:59 and 33 seconds a.m. Eastern Standard Time. Science News: burst of brain activity linked to memory activation. Sleep spindles may help memory storage keep memories separate. Date, May 24, 2018. Source: Northwestern University. Summary: leading theories propose sleep presents an opportune time for important new memories to become stabilized. It has long been known which brainwaves are produced during sleep. In a new study, researchers set out to better understand the brain mechanisms that secure memory storage. The team from Northwestern and Princeton University set out to find more direct and precisely timed evidence for the involvement of one particular sleep wave known as sleep spindle. In the study, sleep spindles, described as bursts of brain activity typically lasting around 1 second, were linked to memory reactivation. The paper, Sleep Spindle Refractoriness, Segregates Periods of Memory Reactivation, will be published May 24th in the Journal of Current Biology. The most novel aspect of our study is that we found these spindles occur rhythmically, about every 3 to 6 seconds, and this rhythm is related to memory. 3 experiments explored how recent memories are reactivated during sleep. While volunteers took an afternoon nap, sound cues were surreptitiously played. Each was linked to a specific memory. The researchers' final experiment showed that if cues were presented at opportune times such that spindles could follow them, the linked memories were more likely to be retained. If they were presented when a spindle was unlikely to fall, the linked memories were more likely to be forgotten. One particularly remarkable aspect of the study was that we were able to monitor spindles moment by moment while people slept. Therefore, we could know when the brain was most ready for us to prompt memory reactivation. If the researchers reminded people of a recently learned fact, a spindle would likely be evident in the cerebral cortex, and memory for that information would be improved. In memory research, we know it is important to segregate experiences while you are awake so that everything doesn't just blend together. If that happens, you may have difficulty retrieving information because so many things will come to mind at once. We believe the spindle rhythmically shown here might play a role in segregating successive memory reactivations from each other, preventing overlap that might cause later interference between memories. Ultimately, the researchers' goal is to understand how sleep affects memory under natural conditions and how aging or disease can impact these functions. With that goal in mind, we've helped elucidate the importance of sleep spindles more generally. Scientists said they are on the trail of physiology of memory reactivation. Future work will be needed to see how spindles fit together with other aspects of physiology of memory and will involve other types of memory testing in other species. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018. It is 10:54 seconds a.m. Eastern Standard Time. New insights into malaria parasite. Date, May 18, 2018. Source: University of California, Riverside. Summary: scientists have found that various stages of development of human malaria parasites include stages involved in malaria transmission are linked to epigenetic features and how chromatin, the complex DNA and proteins within the nucleus, is organized and structured in these parasites. Our results provide insights into the connection between genome organization, epigenetic features, and stage-specific gene expression in the malaria parasite. Professor of Molecular Cell and Systems Biology, who led the analysis of the genome organization of 2 kinds of human malaria parasites. Proteins that are known to control these features could be identified as new targets for novel therapeutical strategies. By disrupting these proteins, we could disrupt the chromatin structure and thus disrupt the life cycle progression of the parasite, ultimately controlling the spread of malaria. The development of malaria parasite through its life cycle is mostly controlled by how the chromatin structure, together with epigenetics, modulates gene expression. Epigenetics involves genetic control by factors other than an individual's DNA sequence. By switching genes on or off, epigenetic changes determine which genes are transcribed. We also found that the epigenetic features are essential and connected with transcriptional activity of several parasite-specific gene families, such as those involved in pathogenesis, liver cell invasion, and regulation of sexual differentiation. Scientists demonstrated that genome organization is not only important in human tissues and complex organisms, but can also affect the normal development of eukaryotic pathogens such as malaria parasites. Malaria, which infects hundreds of millions of people worldwide and kills more than 450,000 people each year, is caused by one of 5 parasites of Plasmodium species. Understanding the mechanisms involved in gene regulation during the various life cycle stages the parasites is important for developing novel strategies to block parasite replication and transmission. Scientists analyze the genome organization when the parasite replicates inside red blood cells and during sexual differentiation in P. falciparum, as well as during the transmission stage from mosquito to humans in P. falciparum and P. vivax. P. falciparum is the most common and deadly parasite. P. vivax, responsible for significant disease, is found in Southeast Asia. Understanding how the transitions between the various life cycle stages of the parasites are regulated remains an important goal in malaria research. The researchers used next-generation sequencing at UCR to understand the epigenetics feature and how the Plasmodium chromatin is organized throughout the parasite life cycle. The sequence showed us how the Plasmodium genome is organized. The Center for Infectious Disease and Vector Research at UCR, we observed that genes that are stage-specific and important in pathogenesis are close to each other. When they are not needed, they cluster together in a repressive center in the parasite nucleus. When they are needed, the chromatin opens up and genes move away from the repressive center. The findings bring a new level of insight into gene organization and dynamics during Plasmodium life cycle, and open up new avenues for targeted approaches toward understanding parasite gene regulation. Molecules inhibiting the restructuring of genome throughout the life cycle have the potential to act as potent anti-malarials. Understanding the genetic and epigenetic elements that control parasite development, including the stages that are responsible for transmission of the disease, is key to the ultimate development of new therapeutical and transmission blocking strategies. A new generous funding grant will allow us to develop a state-of-the-art approach to characterize proteins and molecular components that regulate the gene expression and 3D genome architecture of P. falciparum throughout its life cycle. We expect our new study will offer fresh insights into the biology of the parasite and help us identify novel targets to kill the parasite. Good day, this is Professor Will, CBMD, PhD. Today is Sunday, May 27, 2018, 10:31, 16 seconds a.m. Eastern Standard Time. Science News. Joint resolution, a link between Hutchinson's disease and rheumatoid arthritis. Epigenetic analysis may lead to other unexpected findings and novel therapeutical drug targets for many diseases. Date, May 15, 2018. Source, University of California, San Diego. Summary: using new analytic tools, researchers have decoded the epigenetic landscape for rheumatoid arthritis RA, a common autoimmune disease that affects more than 1.3 million Americans. Scientists said an unexpected connection between RA and Hutchinson's disease opened up the possibility of new therapeutical targets and drugs for both conditions. We did not expect to find an overlap between rheumatoid arthritis and Hutchinson's disease, but discovering the unexpected was the reason that we developed this technology. Now that we have uncovered this connection, we hope that it opens a door for treatment options for people living with either disease. RA is a chronic inflammatory disorder that causes pain and swelling in joints. As an autoimmune disease, it can also affect other organs, including heart and blood vessels. Treatment for RA has improved, but 10 to 20% of patients do not respond to any available medicines. The investigative approach used by the research team involved developing a novel algorithm or a set of computational rules called EpiSig, which integrated and reduced the number of epigenetic combinations in the genes of patients with RA. The team could then identify new cell signaling pathways. Comparing different types of epigenetic data is difficult because it involves a variety of different data subsets that cannot normally be analyzed together, including various methods in which DNA gets modified. This methodology can also be used to find connections between other diseases. As genes involved are discovered, researchers can potentially identify new treatment options and even repurpose existing drugs. Scientists studied the epigenome, in which cells from the joints of patients with RA. Patients with osteoarthritis, which is a disease of cartilage degeneration, served as control group. Both data sets were analyzed through an expansion process that examines chromatin, DNA, and histone modifications. The results produced 12 terabytes of data, 12 trillion bytes, that were then analyzed using Episig. Epigenetics, or above the genome, is a study of processes that alter the gene structure without changing the DNA sequence itself. These DNA modifications are essential to human growth and development and change throughout people's lives. Epigenetic changes are influenced by a variety of environmental factors, including stress, activity, and life cycle choices. Researchers say by revealing the comprehensive epigenetic behind rheumatoid arthritis, we now have a better understanding of this disease. More importantly, our new approach could not only help patients with rheumatoid arthritis, but also people with other immune-mediated diseases. Good day, this is Professor Will, CBMD, PhD. Today, Sunday, May 27, 2018, is 10:42:57 seconds a.m. Eastern Standard Time. Researchers discover mechanisms and epigenetic markers with implications for disease ranging from cancers to infertility. Researchers open the door for future stem cell research in chicken models. Date April 30, 2018. Source, University of Maryland. Summary. A researcher uncovered new mechanisms that dictate the development of germline stem cells. Mechanisms were found to be associated with genes responsible for cancers and viral infections, among other major diseases. Markers used to identify male germ cells were discovered, exploring how environmental factors or epigenetics affect these cells and providing significant insight into treatments for male infertility. Findings set the stage for chickens as more prominent model organism for stem cell research. Stem cell research has applications for treating cancer, heart disease, neurological disorders like Alzheimer's disease, diabetes, and even injuries. These cells can replicate and replace damaged tissue, so understanding their development is important to the future of this work. Epigenetic factors, or environmental factors that affect the way genes are expressed, can also play a large role in the cell and tissue development, and no studies have previously looked at the mechanism at play in germline stem cell development and how epigenetics play a role. From genome to phenome, as it is called, is a very complicated process for different genes networks to create the trait you see in an animal or human. The activity of key genes is the same or similar in common cell types, but the activity of other genes may differ completely among species, especially for the roles of stem cells. Looking at these mechanisms helps to decode genes, understand complex traits, and develop future treatment plans to better understand animal and human health. How was the focus of the study, examining germ cells to clarify mechanisms that factor into germ cell development and gene expression. Specific processes were discovered for individual genes with associations to cancers and viral infections, and markers used to identify and track genes were discovered in male cells with implications for infertility treatments. These epigenetic markers are unique to chickens, which is an up-and-coming animal model that is in many ways ideal for the study of epigenetic stem cells and developmental research. Most people still think of mice when they think of animal models that support animal and human health research, but regulatory elements are quite similar between the chicken and human genome. In fact, the genetic similarity between a human and a chicken is about 60%. Development of chickens is rapid, easy to see, and easy to manipulate, making them very unique compared to other animal models. They're ideal for developmental and stem cell research because you can easily observe egg growth and the development process in real time. Researchers not only devoted to this work with animal genetics and connections to human health in mind, but is also thinking about the broiler chicken industry and overall animal welfare. This work provides a deeper understanding of developmental mechanisms in chickens that lead to healthier chickens and humans. The application and need for epigenetic stem cell research is great, and chickens are a great model for this work.